We just finished learning about the diversity of plants and animals, and I want to point out that animals have been around since the Precambrian. Now, many groups arose in the Cambrian explosion, and we had full diversity by the end of the Cambrian. And then plants invaded the land in the Paleozoic, and they're both experiencing similar environmental pressures as they invade the land. And we talked a bit about that when we talked about the different adaptations that groups of plants and animals evolved in relationship to their colonization of the land. The course now is going to take a turn and look a little bit at those adaptations and those important tasks that every living thing has to carry out in order to survive, and the organs we've evolved to help us accomplish those things. So I'll call this section Animal and Plant Anatomy and Physiology. Being alive is a very difficult task. You have to maintain internal conditions separate from those outside. So you must maintain homeostasis, you must survive predation, you must find food, you must get rid of wastes, etc., etc. It's very difficult. And one very obvious sign of this is the way that organisms have adapted to their own particular environments, especially in harsh environments. It shapes the way organisms look and behave. So the two plants on the left are very distantly related, but they both live in harsh desert environments. They've adapted similar body plans due to convergent evolution. The organisms on the right are all very, very distantly related. We have mammals, birds, and fish here, but they all live in similar habitats and have evolved due to convergence similar body plans. So analogous features have arisen, making them well adapted to swimming. Of course, similar adaptations take place internally, which is why we must study not just external morphology but internal anatomy to understand how organisms are adapted to life on the planet. In lab, we looked at this organism, Paramecium multimicronucleatum. It's a single-celled organism that swims around with cilia. How does this organism get all the food it needs, all the materials it needs to survive? I think you know this. It's basically through diffusion. Substances like water and oxygen and food are taken in. Food through phagocytosis, water through essentially osmosis, gases through diffusion. So oxygen is coming in, water is coming in, etc. But other items like CO2 and other wastes have to be excreted and gotten rid of. But note that this organism is one cell big, so it is entirely in contact with its environment. It can directly interact with the world. Now, when we get to become multicellular, our cells become much more complex, and not very many of our cells are actually in contact with the world around us. Many of them are deep within our bodies, where they cannot directly react with the environment. Because of this, we have evolved tissues and organs in order to make up for this fact that our cells are no longer in direct contact with that environment. So nutrients, waste gases, etc. must get across cell membranes in order for the cells to survive. So our cells still need all the same things that paramecium needs, but our cells are now in this huge trillion cell big gigantic organism that is a human being, and they must still do these things. They must get gases in and out, they must get waste out, they must get nutrients in, all across those membranes. And one thing that you will know by now is that surface area to volume ratios are imperative to fully understand anatomy and physiology. And the rate of exchange between the cell and its environment is directly correlated with its surface area versus its volume. And cells must have a lot of surface in relation to their volume to get enough nutrients in, get enough waste out, etc., to survive. So how can a huge organism like you, with cells that are way deep in the middle of it, and cells that are scattered throughout, how can they possibly get enough oxygen? How can they possibly get rid of their waste? How can they possibly get enough food to survive? And of course, we know that that answer is organ systems. Now, when we look at single-celled organisms like this amoeba or like the paramecium, they directly exchange with the environment. But then, if we focus on animals, we go to a simple multicellular animal like this hydra. Note that in simple multicellular organisms, all of the cells are still in direct contact with the environment. So notice the hydra can exchange gases and food with the environment out here, as well as within that gastrovascular cavity. So it's multicellular, it's fairly complex. I say simple, it's a fairly complex organism, but its body plan is quite simple in that all cells are still in direct contact with the environment. But then you get to organisms like flatworms all the way up to dolphins and elephants and things like that, and we have lots and lots and lots of different organs that have evolved to accomplish all of these tasks. Not just organs, but organ systems. Many different organs made of many different tissues, all working together to accomplish what an amoeba can do just in its own environment. But our cells cannot do in their own environment, because their environment is the center of a heart or the center of a lung or the center of your brain. They cannot just pollute the environment around themselves with waste. They have to get it out of the body in order to avoid killing themselves. So first of all, we've evolved highly branch and folded structures, which means structures with tons of surface area to allow for rapid exchange of materials. This includes all of those organ systems that you may be familiar with, and if you're going into the medical field, you will have to become intimately familiar with. Whether it be the digestive system, as we see here, this organism has this is just a model organism, it's not a real organism obviously, but it has a mouth and it has a digestive system, and we can see these folded regions of the digestive system shown here, extensions of the small intestine that increase surface area, so we're able to absorb nutrients that way. But notice that's not the end of the road. The nutrients have to get to your cells. They do that by diffusing or by being pumped into a circulatory system. So the circulatory system now allows those materials to be sent throughout the body to all the other organ systems. So the cells all throughout the body lie within close approximation of some of these blood vessels, so they can absorb nutrients and undergo basically endocytosis and then use those materials in their mitochondria, etc., etc. That blood also goes to a respiratory system where gases are exchanged. Remember, an amoeba or a paramecium does this directly with its environment, but our cells don't have that luxury. They're buried deep within us. So we have a circulatory system that pumps blood to a respiratory system where gases are exchanged and then distributed throughout the body. This is accomplished with a highly evolved lung tissue, in our case, that has tons of surface area in relation to its volume. Then that oxygenated blood goes throughout the body, and as we see here, exchanges take place with cells. It's a two-way exchange. CO2 is taken up from cells, waste is picked up from cells, and nutrients and oxygen is delivered to those cells. So then our blood is full of waste, and it has to be filtered, and that is what the excretory system is for. Waste is pulled out of the blood and then released as metabolic waste, such as urine. And then the blood continues to go through the rest of the loop through the body. So all of these organ systems are doing the same thing that just a simple paramecium does all on its own. But because that paramecium is in direct contact with its environment, it doesn't have to have all these organ systems. Obviously it can't, it's a single cell. But our cells have to have all of this support in order to survive. So organs are composed of tissues. So remember, tissues are composed of cells. So you'll have specialized cells living together making a tissue. Those tissues come together to make organs. So you might have more than one tissue in an organ. You might have more than one cell type in a tissue. Then you have many organ systems working together. So you have tissues, organs coming together in organ systems. What I mean by a system is like the digestive system is composed of different organs like the intestines and the stomach, etc. But then you get many organ systems and you put them together, and that's when you get a true multicellular organism. So an organism is a living thing composed of organs. Is technically what that word means. And now that we have all of these systems in place, now these systems evolve slowly, and some organisms have fewer of these systems and some of us have more of these systems, but that allows us to maintain internal conditions and to utilize negative and positive feedback in our lives to maintain homeostasis. The more common one of the two in our daily lives is negative feedback. Negative feedback, let's say your blood sugar is low. You will feel hungry, and that'll cause you to eat, causing your blood sugar to go up. So the stimulus was low blood sugar, and the response is raising your blood sugar. That is what it means by negative feedback. Just like when your apartment gets cold, your thermostat turns on the heater and it warms it back up. So the stimulus is in the opposite direction from the response, basically, negative feedback. The stimulus is in one direction and the response is in the other. Positive feedback is much more rare in our cases. Childbirth is one instance where we see positive feedback, where contractions cause more contractions to happen. So in positive feedback, the stimulus and the response are in the same direction. Positive feedback. Now let's look at the world of plants. That's where I want to spend the bulk of the rest of this time, because we're going to spend a lot more time with the animals since we have more organ systems. Let's talk about the major organs of a plant. The major parts are not too difficult. We have roots, shoots, and leaves. So think of shoot. Technically, I should say we have roots, stems, and leaves. A shoot is technically stems and leaves combined. So the root is at the tip of the plant. It's an organ that is involved in absorbing nutrients and water. And the root grows at the tip. And this is the reason why we used the tip of a root when we looked at mitosis slides in Bio 1. So the root has a root apical meristem. So this area down here is called a meristem. We've heard that word before. It's basically an area of undifferentiated cells that divide over and over and over again, causing the root to elongate. And as those cells back here mature, they mature into xylem and phloem, and root hairs form, things like that. So root hairs, we've already talked about before, are these tiny little extensions of epidermis that allow for increased surface area, allowing those roots to absorb more water than they could otherwise. So at the tip down here we have the root apical meristem, and up here we have all of these root hairs. I would write it down, but I just have a mouse right now. I don't have a pen. And another way to increase the surface area of roots is mycorrhizal associations, as we've discussed, symbiotic relationships with fungi. Meanwhile, stems are also growing, and they grow at the tip, and the tip of a shoot is called the shoot apical meristem. It's found at the very top of the shoot, and the growth of a stem then is it's built in sections, repeating units over and over and over again that we call nodes and internodes. So a node is basically what we call like right here where this bud is, it's a node. And then the areas between the buds are called internodes. So a bud, like we see right here, right, this is a bud. This is a shoot apical meristem. It can grow into a new branch. And they're always found in what we call the axil of the leaf, which is just the upper angle of the leaf between the leaf and the stem. That's where you find buds. So nodes are where leaves are attached, if we want to define it, and internodes are between where leaves are attached. And you'll just see that repeating over and over and over on a stem. So we have a terminal bud, the shoot apical meristem, found on the tip of the plant. That's the main bud that divides over and over and over again, causing the shoot to get longer. Axillary buds are these buds on the side of the stem, like the one I circled down here. Those give rise to branches, or they might give rise to flowers, and they're found in the axil of the leaf. The words are covered up by the picture here. The axil of the leaf, which is just that angle of the leaf between the leaf and the stem on the upper side. So if we zoom in on a shoot apical meristem, we'll see something that looks like this. This is not Batman. This is the shoot apical meristem, and right in this area is where mitosis is happening. So right in here. And as you move down this way, we're seeing older cells. So the newer cells are right here, older cells are down here, and what we see right here, it says leaf primordia. That means it's a really young leaf. And look, down here is a much older leaf. We can see just part of it over here. And we also see buds forming. So axillary meristems. So those are the buds right there in that crook of the leaf. So this plant is going to grow from that apical meristem. It's going to elongate. So this is going to get longer. And we're going to just repeat these units over and over again. It's going to make more leaves and more leaves and more leaves, and we'll get this node and internode kind of growth form. So when we look at elongation in plants, whether the roots are growing down by mitosis at the root meristem or the shoot meristem is a little more complex. It makes leaves and buds as well as grows longer, okay? But the stem is growing longer, the roots are growing longer. This is known as primary growth, okay? Primary growth. I could try to write it with a mouse. It probably won't be pretty. Oh boy. Mary. That's awesome. Anyway, I'm not gonna do that. Primary growth, that is getting longer. Secondary growth in plants is getting wider around, and we really only see that in things like trees, where we make a lot of wood. So secondary growth involves the production of bark. I'm not going to worry about all these other terms, but bark and secondary xylem. Bark includes the production of phloem and a new periderm, which is like an epidermis. So, and then secondary xylem is the production of a bunch of xylem that every year a new layer is added, which is why you get growth rings in a tree. So remember that secondary growth is essentially, for our purposes, the addition of new xylem every single year, and therefore every single year the tree gets wider and wider around, producing growth rings. Primary growth is elongation. Secondary growth is getting wider due to adding more layers of xylem. The part I want to spend just a little bit more time on is a discussion of leaves, because leaves are one of your key characteristics in identifying plants. And I think it's really important that a student in Bio 2 at least know a little bit more about the world around them, at least the world on campus when they leave than when they came in. So remember, leaves are the site of photosynthesis as well as the site of gas exchange. This, of course, being a leaf of poison ivy. Just giving you another reason why it's important to know your plants so you don't stumble into this by accident. And leaves have a couple of parts. We have a blade, which is just the green leafy part here, and the petiole, which is the stalk right there. And you'll always find the bud, the axillary bud. Axil means that angle between the leaf and the stem. You'll always find a bud right there in the axil. And leaves come in two major types: a simple leaf, where there's just one blade, or a compound leaf, where there is many blades, or leaflets. So many leaflets. How do you know that this is not 1, 2, 3, 4, 5, 6, 7 leaves. How do you know this is not a 7-leafed branch? Well, there's not a bud down here on these leaflets. There's only one bud, and it's down here where the petiole, which is the stalk of the leaf, meets the branch. So there are simple leaves, once again, like we see there in the oak, and there are compound leaves, like we see below. Something like an ash tree or a pecan tree. Pecan tree will have compound leaves. Leaves can also be heavily modified in other ways. The spines of cacti are leaves. The climbing tendrils of some vines are leaves. And the stuff you eat in an onion is, except for this part right down there, is stem, but the rest are leaves. Those are storage leaves. So leaves can be heavily modified by evolution for other purposes. But their typical structure is this way on most of the plants that we see. Oop, let me go back. So leaves have a very predictable morphology. They have an epidermis, an upper epidermis, a lower epidermis. They have stomata, which are the gas exchange structures. Remember, stomata have guard cells that look kind of like kidney beans. I'll draw it as best as I can with this mouse. So here's 2 guard cells, and then right in here's the pore where the gas exchange occurs. Obviously, the picture is a lot better over here. They have a pore, 2 guard cells. They open and close and let gases exchange. CO2 in and O2 out. And then we have these layers of cells inside known as the mesophil cells. So the palisade mesophyll are these elongate cells here. That's where the bulk of your chloroplasts are. That's where photosynthesis happens in the palisade mesophyll. And then down here you have the spongy mesophyll, which is round cells that are more spread out, lots of air spaces between them, lots of surface area. This is where most of your gas exchange is going to take place. So you should be familiar with that. And then also running through these tissues are vascular tissue composed of bundles of xylem and phloem, transporting water in the xylem and transporting sugars in the phloem. So that's what's in your leaf. That's what they do. They are gas exchange / photosynthesis factories, and that xylem and that phloem runs all the way from the roots, so they label it here vascular tissue, all the way from the roots all the way up to the stems. And in the human body, for example, we have the vascular system that pumps water around, exchanging materials. Plants have their own vascular tissues, xylem and phloem. So when we look at a plant, we can see this major body plan where we have an apical bud. That's where the shoot apical meristem is up here. And then we have lateral buds down here, some of which will develop into flowers, some which will grow into branches. And we have this repeating unit of nodes where leaves are attached and internodes, which is between where leaves are attached. And here we can see where a lateral bud has developed into a branch. And then down here we have our roots, the major root being called the taproot, and then any roots coming off to the side known as lateral roots. So that's the basic morphology of your average plant. The reason I want to talk about nodes and internodes is you often, when identifying plants, need to look at how many leaves are attached at the node. So are leaves opposite one another at a node, or are leaves alternate? So nodes only have one leaf each and they're alternately arranged like we see on this diagram. So like I said, leaves are very important tools for identifying plants. So there are different kinds of leaves. On the left we have a pinnately compound leaf. So you know this is a compound leaf. These are only leaflets, not individual leaves. There's only one bud down here, and this whole thing is a leaf. So this entire structure is one leaf called a pinnately compound. So it's pinnate meaning like feather-like, or think of a feather pen. It's feather-like in its structure. Some are even more complex like this one. I won't get too worried about bipinnately compound, but that's when there's a whole bunch of structures coming off here, but this is all still a single leaf. And then palmately compound leaves are still compound leaves, but there's one bud, but the leaflets all come off of one central place. So they call this palmately compound. Think of like your palm with all of your fingers coming from the palm of your hand. Those leaflets come out that way. So we see this on campus in the form of buckeye trees is a good example. So we talked about simple leaves already. That's where there's just one leaf, one blade and a bud. But compound leaves, like we see on this slide, can come in a few types. The two you should be most familiar with are the pinnately compound and the palmately compound leaf. So in the winter, when you look at a plant, you'll see the twigs like this, and you won't see leaves. You'll see just scars and buds. Remember, buds are dormant apical meristems, whether they're axillary buds on the side or the bud on the tip of the branch. And you'll see scars on the side of the plant where leaves used to be attached, and the buds are always right there in that axil of the leaf where the leaf used to be. So keep an eye for that next time you're out in the wintertime. Now when we're looking at identifying plants, whether it be in the winter or whether it be in the spring, we look to see whether the leaves are arranged with one per node in an alternate arrangement like we see here. One node, one leaf, one node, one leaf, one node, one leaf, and they're alternately arranged, so one goes this way, then one goes that way. If they're oppositely arranged with 2 nodes per leaf, or if they're whorled all in a circle at a node. So there's different kinds of leaf arrangements which will help you to identify different species. I'll just run down 2 common genera. So 2 different genera you may experience on campus. One is the oaks, the oak trees, which is the genus Quercus, and it has a ton of species in Arkansas. It has simple leaves, meaning a bud and a leaf. It produces fruits known as acorns, and the terminal buds on the tip of the branch are actually found in clusters, so you can identify these in the winter as well. The terminal buds have clusters, or are found in clusters. And there are 2 major groups, red oak and white oak. I won't get too worried about that, but red oak tends to have pointed leaves. White oaks usually have rounded leaves. So they have alternate simple leaves, and if we look at their leaves, we can see that they look like oak leaves, oddly enough. The weirdest one that I know of is the one found all over campus, which is willow and water oak, which has leaves that look like this. They're highly derived, but most of your oaks have leaves like those on the right. Lobed white oak leaves or pointy red oak leaves. They will be alternately arranged, simple leaves, and the buds on the tip of the branch as well will be clustered like that. So how do you tell that from a maple tree? Well, the reason I picked these 2 is almost everyone's at least heard of maple and oak. Maple trees have opposite simple, and I say usually simple, there is an exception, but most of them are simple. But the leaves are opposite, so that's a lot different than an oak. The buds are not in clusters. The fruit, unlike an acorn, are these things called samaras, or you might call a helicopter. So these are also all over campus, but easy to tell apart from oak. So I want you to practice that while you're walking around to classes. Try to pick out a few oaks and a few maples as you walk around. So that was a brief overview of a little bit of physiology and a little bit of basic plant anatomy. We're also going to move into animal anatomy and organ systems, and then I'll do a lot of side-by-side comparisons of plant and animals rather than doing entire lectures on one or the other.